Hello, biology students. Are we ready for Chapter 7? Photosynthesis is a confusing topic, so hopefully this video will help explain some things, because if you're just trying to read it in from a book, it can be confusing. But I've gotten pretty good at explaining it over the years, so hopefully this will be helpful to you. Another thing I highly encourage you is there are a lot of good YouTube videos out there about the process of photosynthesis that shows good graphics and good movements of different things. And so it would be really helpful to just Google or put in YouTube photosynthesis, and you should be able to come up with some really good videos, because this is confusing. All right, photosynthesis. We're going to talk about the organisms, the process, how they convert solar energy, fixed carbon, and other types of photosynthesis. So all life on Earth depends on the sun. Again, there's a few exceptions that are very extreme, the chemosynthetic organisms that live in the ocean, but for the most part, it depends on the sun. Photosynthetic organisms are the ones that are green, such as algae, plants, and cyanobacteria. They take the solar energy and turn it into chemical energy. Now, we learned in the last chapter that we can't really use photo energy as a consumer, but we can get that sugar from a plant, and the plant does take that solar energy and turns it into a chemical energy that we can use. So autotrophs are organisms that can make their own food and produce their own food by photosynthesis. Photosynthesis is a process that captures the solar energy, turns it into a chemical energy, and actually makes carbohydrates, which can be stored and broken down later through cellular respiration. Photosynthesis produces food energy. Now, it does produce a little bit of ATP, but most of the ATP production takes place in cellular respiration. Really, what photosynthesis is producing is mostly going to be those sugars. And it makes those sugars by a combination of carbon dioxide and water, and taking in carbon dioxide and water and rearranging them into sugar and oxygen. So this is very important for us as heterotrophs. As heterotrophs, we need to eat in order to get our energy, and we depend on the autotrophs, the ones that are making the energy. Now, photosynthesis takes place in the chloroplasts, and the chloroplasts are the green structure. Now, there's a whole reason that they are green. It has to deal with photosynthesis happens better on certain spectrums of light than others, and the green is reflected back because it is not as useful for photosynthesis. Anything that you are seeing with light is actually reflecting back to you. And so if you are looking at the screen right now and seeing red, that's because the red color is reflecting back to your eye. So if we're seeing green, it's because the other wavelengths of light are more helpful for photosynthesis, and green isn't. And so green is reflecting back to us. The raw materials for photosynthesis are carbon dioxide and water, and they go into the reaction and get recombined in order to make the sugars and the oxygen. Roots absorb the water, and it moves up through the vascular tissue through capillary action, and carbon dioxide enters through the leaves in openings. So at the leaves of plants, there are tiny, tiny openings called stomata, and the the plants can control how many stomata are open or not. Typically, they don't open the stomata a lot during hot days, because they also lose water through those stomatas, and they want to conserve water on hot days. But the stomatas are cells that are little openings that allow the carbon dioxide to diffuse in, because we talked about diffusion as well. Diffusion and osmosis are going to play a big part in this photosynthesis process. Now, where this is all taking place is inside the chloroplast, and inside the chloroplast we have something called a thylakoid membrane. The membranes and that process of going across the membrane by diffusion and osmosis, that's where our energy is going to be created. So these processes actually take place across a membrane. In this case, the membrane is called a thylakoid in the chloroplast. Now, also within the chloroplast, we have something called the stroma. The stroma is like the cytoplasm of the chloroplast. It's like the jelly kind of material that fills in the empty space of the chloroplast. So we've got our thylakoid membrane, we've got our stroma, and all of this takes place inside the chloroplast. So here's our chloroplast. Our chloroplast has two membrane layers. We talked about that before, how this was some organelle that might have gotten engulfed by a bigger cell. And we see that we have these thylakoids that look like pancakes stacked on top of each other. All of this surrounding fluid, that's the stroma. And there will be some processes that take place in the stroma. But most of these processes are going to take place across this membrane of the thylakoid. So here we see how this whole autotroph and heterotroph thing works. We have the solar energy coming in. The autotroph is making the glucose and the oxygen. This gets sent to the heterotroph when we eat it. It's not like really sent to us. We have to eat it. And then cellular respiration creates that energy. Carbon dioxide and water are produced, and that gets reabsorbed by the plant along with sunlight. Process repeats. So there are two main processes that take place in photosynthesis. The first is called the light reactions, and they only can take place when light is present. This process does require that input of energy. So there are energy capturing reactions, and the chlorophyll is going to absorb this sun energy, and the electrons move down a transport chain. Again, think back to those electrons being very important with energy bonding, allowing like the different shells. All of it comes down to energy with the electrons. The electron transport chain is going to do something called pumping hydrogen ions into the thylakoids. So this goes back to what we learned about with diffusion. We're trying to get a high concentration of hydrogen on one side of this membrane when that's typically not what a cell is going to want to do. And we're pushing these hydrogens to one side by using electrons. The electron transport chain is used to make ATP out of ADP. Also these things called NADPH and NADP. These get used again, another important thing to remember. With photosynthesis and cellular respiration, nothing is really wasted. Everything has a purpose. We are going to see this NADPH and NADP plus come back again and use this again. There is a purpose to everything that happens in these processes. Next, we have the Calvin cycle, which takes place in the stroma. The Calvin cycle, its job is to take the carbon dioxide and reduce it to carbohydrates. Combining that carbon dioxide with hydrogen ions, here's some hydrogen ions, to make the carbohydrates, again, which was C6H12O6. It uses that ATP, and then there's that NADPH that gets used to make those carbohydrates. Reaction was named after Melvin Calvin, who used the carbon isotope to trace the carbon in photosynthesis. Honestly, if you think about how complicated this process is, and somebody actually figured out the step-by-step part of it, I mean, it's hard enough to understand, no less to figure it out for the first time. So this Melvin Calvin guy, he deserves to have something named after him. So photosynthesis involves oxidation and reduction, and we already talked about the gain of electrons and the losing of electrons. And photosynthesis, carbon dioxide is reduced and water is oxidized. So here we can see just a plant. This is like a water plant, and it's got water bubbles because oxygen does get released as a waste product. So this plant is making oxygen bubbles because it is going through photosynthesis, and photosynthesis releases oxygen. So bubbles will form on a plant just magically underwater, but it's not magic. It's because of photosynthesis. So here we can see a chloroplast. The chloroplast has the thylakoids and the thylakoid membrane and the solar energy that is coming into it. Water also coming into that light reaction, oxygen being reduced, the NADPH and ATP being reduced, and we have ADP going in because this is going to be used for ATP, to make ATP, and NADP plus also going in. So we have these products going in, these products coming out. Then we send this into the Calvin cycle. This does not require light to happen. Only this process needs light. This process does not need light. It happens in the stroma and no light energy is required. What it does use is this ATP and this NADPH. These two things are used for this process. Also, this is where carbon dioxide comes in, and we have CH2O produced, which will later be turned into C6H12O6, which of course is glucose. So very simplified summary. Solar energy coming in, water coming in, ADP and NADP plus coming in. Then we have oxygen being produced. NADPH, ATP going into the Calvin cycle. Calvin cycle takes in carbon dioxide as well, happens in the stroma, and eventually sugar is produced. But again, that ATP is used up and that hydrogen is taken off here, so those two can be sent back to the first process. Nothing is wasted. Everything has a purpose and everything can be reused, meaning this is a very efficient system. It's a complicated system, but it's very efficient too. Okay, so absorption spectrum, pigments and photosynthesis. Chemicals that absorb certain wavelengths of light are pigments. And so wavelengths that are not absorbed by pigments are reflected or transmitted. So this is what I was saying earlier. The reason that we see green is because it is not absorbed by the chloroplast and it's reflected back to us. And so it's using the other colors in the photo spectrum and it's not using green. Green is not helpful to it, so that's why it's reflecting it back. So the absorption spectrum, the pigments found in chlorophyll absorbs various portion of the visible light because light can be broken up into wavelengths. And it shows that chlorophyll is green because it absorbs much of the reds and blues of white light and reflects the green. There are carotenoids as well, which are accessory pigments, and these carotenoids absorb light in the violet, blue, green range and reflect yellow and orange. When do we see yellow and orange leaves? During fall. So the yellow and orange gets reflected in the fall because it is not using that as often, and we're switching to these other pigmentations to try and get a little bit of last bit photosynthesis. Typically the chlorophyll is what we are seeing, but carotenoids are being seen in fall and the yellow and orange light is being reflected. Here's an example of that wavelength. So this is all we can really see. There's lots of other wavelengths available for light. We can only see this tiny bit. But I'm sure you've heard of X-rays and gamma waves and microwaves and radio waves. That's all light waves, with radio waves being further apart and gamma rays and X-rays being more condensed and shorter. Shorter waves, more energy, by the way. Longer waves, less energy. So we're right here in the middle. And if we're looking at chlorophyll, chlorophyll is on the red-blue spectrum and also in this orangey-red spectrum. Not much chlorophyll being needed for the green spectrum, so that's why that gets reflected back and that's what we see. Then we have our carotenoids. Carotenoids are starting to absorb all of these, including the greens, so the only thing the carotenoids are reflecting back are the yellows and the reds, which is why the leaves again turn yellow and red in the fall. Now, with photosynthesis, we have two pathways. We have the cyclic pathway and the non-cyclic pathway. One of them just means it goes in a circle because a cycle is a circle, and one of them does not go in a circle. And a plant can alternate between these based on its needs, and some plants will use one a lot more than the others. So light reaction captures light energy. There are photosystems 1 and 2. Kind of weird. Even though it's photosystem 2, it comes first in the process, and photosystem 1 comes later because photosystem 1 was discovered first in the cyclic pathway, and photosystem 2 was discovered later, and so that's why they're like flip-flopped. So photosystem 1 and photosystem 2. And a photosystem is just like where the light hits to collect pigments. So think of it as like an antenna that's collecting the pigments. So as the sun is shining on the plant, the pigments are going to these photosystems. Both cyclic and non-cyclic pathways produce ATP, but the non-cyclic pathway also produces NADPH. and this will go on to the second part of photosynthesis. So I know this all kind of sounds confusing, and again, this is why it is helpful to go back and watch those videos on YouTube as well, because these processes get explained a little bit more. So the non-cyclic pathway takes place in the thylakoid membrane, okay? And it uses photosystem 1 and photosystem 2. Both are used in the non-cyclic pathway. Photosystem 2 captures the light. Again, it starts with photosystem 2, even though photosystem 1 sounds like that should come first. It starts with photosystem 2. It captures the light. Begins with photosystem 2, and it causes an electron to be ejected. So now we've got this electron bouncing around. Think of the electron as a bouncy ball. When those little tiny rubber bouncy balls you used to play with. You throw that bouncy ball, it's going to bounce off a wall. It's going to bounce off the ceiling. It's going to bounce off the floor. Eventually, it runs out of energy, but it's going to bounce around for a while. That's what happens with these electrons. These electrons are bouncing around, and every time it bounces, energy is created. So if we can kick that electron and get it bouncing, we can use the energy that that electron is bouncing around with. And so that's where that electron transport chain comes in handy. The electron transport chain is just that electron bouncing around, giving us some energy that the plant can utilize for the reaction to happen. Eventually, these electrons get replaced with an electron from water. And so when we use up one electron, we can get another because we've got lots of water. And so we're going to break apart that water. And we've got oxygen. Hey, look, oxygen, isn't that a byproduct of photosynthesis? So here's where our oxygen comes from. We split apart that water, and now we've got some hydrogen. This causes hydrogen to accumulate in the thylakoid chambers. Again, not what the cell wants, but in this case, we're trying to get a lot of hydrogen concentrated on one side. This time, we want the hydrogen. Came from the water, we split the water apart, we got that electron. That's pretty cool. And now we've got oxygen. Hey, we don't need the oxygen right now. We will let that go away and use it for cellular respiration later. The hydrogen gradient is used to produce ATP. This comes later. Eventually, all that hydrogen is going to build up and build up and build up until it flows back through the membrane through a special protein that creates ATP. Then, after all of this happens with photosystem 1, then we finally get to photosystem 2. Photosystem 2 captures the light energy and gets rid of another electron. So now we're bumping that electron again. It's going to give us a little bit more because that ball is bouncing around a little bit more. The electron is transferred permanently to a molecule of NADP. So now it causes NADPH. Photosystem 1 is making this NADPH. All right, it looks confusing, but let's take it step by step. If you take it step by step, it's not so bad. You can do this. Solar energy comes and hits our photosystem. Photosystem is going to break apart that water. We got an oxygen. We also get an electron. The electron bounces, bounces, bounces like that bouncy ball thrown around the room. This causes hydrogen to come over that membrane and start to gather down here, and it's allowing us to create a little bit of ATP. By the time we get to photosystem 1, more energy coming. This bouncy ball of electron, it's out of energy now. But it can be put into this photosystem with a little bit more light, bouncing a little bit more, and then we can attach that hydrogen to make NADPH. This NADPH, along with that ATP, it's going to go into the next step. So this is going to be used later. ATP going to be used later. Goes into the Calvin cycle, which is the second step. All right. So photosystem 2 consists of a pigment complex, just like it needs sunlight. Oxygen is released, splits water, all that we've seen before. The electron transport is going to bounce around that electron, carries the electron from photosystem 1 to the photosystem 2, and pumping those hydrogens through. Again, we're concentrating these hydrogens. We want lots of hydrogens, not normal, but in this case we want it. Photosystem 1 has the pigment and electron acceptors. We make an NADPH from an NADP. And this is where the electrons go. Then we have what's called an ATP synthesis. We've got a high concentration of hydrogen now. All those hydrogens we've been pumping across that membrane by bouncing those electrons around. What are we going to do with them? Well, because of diffusion. Remember diffusion, things go from a high concentration to a lower concentration. Those hydrogens want to flow back out, and they are, but they're going to flow back out through this ATP synthase. Anytime something ends in ASE, that means it's an enzyme. We didn't talk about that yet, I don't think. But synthase means that it's an enzyme, which means we are going to create ATP. The hydrogen that's concentrated is going to flow through this ATP synthase. It causes it to spin like a wind turbine. And when it spins, that spinning motion creates energy, just like wind creates energy when the wind turbines spin. Or like when water is going over a wheel, the water's causing the wheel to spin and energy is created. The hydrogen ions flowing through this ATP synthase is going to cause the ATP to spin, or the protein enzyme to spin, and ATP will be created. All right. Confusing? Yes. But possible. Here is the cyclic pathway. What we were going over before with all of those steps was the non-cyclic pathway. With the cyclic pathway, if we wanted to just get some quick sugars, then we would have the photosystem 1 with the electrons that we get from water, energy from the sun, the electrons bounce around, and they come back to photosynthesis 1. So it's a cycle. It's a cycle. Eventually, you can send some more stuff to that Calvin-Benson reaction, that Calvin cycle. But the cyclic pathway, it's one way. Okay, this is the whole process. So let's take it step by step. Thylakoid membrane in a chloroplast. Okay, here's our thylakoid. Okay, thylakoid membrane. Photosystem 2. Light energy comes and strikes it. We've got a water. We don't necessarily need a water, so we're going to break it apart. We're going to release some oxygen. This oxygen goes away out of plant, so we can use it for cellular respiration. Then we've got these hydrogens that are left over. H2O, we've got hydrogens left over. Lots of hydrogens in here. Not normally what the cell wants. But we've got this electron that we got from the water. We're going to send it bouncing through that electron transport chain. That's going to create a little bit of energy. Let's push some more hydrogens in here. We've got some hydrogens from the water, but let's put some more. That we can get that energy from that electron. Sends it to photosystem 1. With photosystem 1, we've now got some more light coming in. We can take that electron and we can attach it to an NADP plus and make an NADPH. The NADPH will go to the next step, the next process. Okay, great. Kind of done with this, although this process kind of keeps going and going and going. What are we going to do with all these hydrogens that we have here? Well, here's our ATP synthase, the special enzyme. These are going to naturally diffuse out because high concentration diffusing to a lower concentration. They're going to naturally go through this ATP synthase, and as they do that, it causes it to spin, and ATP is created. So finally, we've got some energy. This is where energy is created. This energy, though, and the NADPH, those are both going to go to the Calvin cycle because we need to make sugars. This is only the first step, is to make the energy and the NADPH. Now we need to make sugars. It would be advisable to go back and watch me explain that portion again because that is going to help you understand the process. Water's broken, using light energy, electrons bouncing around, hydrogens getting concentrated by electrons bouncing around, going through that synthase, making energy. All right. So ATP production, the thylakoid space acts as a reservoir for those hydrogens. Each time a water is broken, hydrogens are left over. Those electrons bouncing around, and more energy is going to be created. And so that pumps even more hydrogens in. The hydrogens, again, they don't want to be concentrated. That's not normal, so they're going to flow out naturally through that ATP synthase. And as they flow out, the ATP synthase is going to spin, and it makes the ATP from the ADP. This is called chemiosmosis when this process happens with the hydrogen gradient. Okay. Next step is plants fixed carbon dioxide. This is the next step that is going to utilize the carbon dioxide and turn it into carbohydrates. So this is a cyclic series reaction, so cycle, okay, go in a circle. And it takes carbon dioxide, that's where our carbon comes from. Until now, we haven't had carbon. We just had oxygen and hydrogen and some electrons. We haven't had carbon, but we get our carbon from the carbon dioxide, which we're absorbing. C3 photosynthesis, and it involves 3 stages: carbon dioxide fixation, carbon dioxide reduction, and Ruby-P regeneration. This can also get a little bit confusing, but as long as you are understanding what goes in and what comes out, those are the main parts of photosynthesis. If you want to learn more, and this is interesting to you, then you can take like a botany class and it goes more in depth. But really just try and focus on what goes in and what comes out. First of all, we have carbon dioxide coming in. It attaches to something called Ruby-P, which is a carboxylase ACE enzyme, ASE enzyme. Results in a 6-carbon molecule, so we're having 6 carbon dioxides coming in to make a 6-carbon molecule. This splits into a 3-carbon molecule. Reaction is accelerated, and then CO2 is now fixed because it becomes part of a carbohydrate. So again, the cyclic path of this Calvin cycle, we're taking the products that we used, or the products that we gained from the first light-dependent reaction and putting them into the light-independent reaction. So we've got the 3 carbons coming in, and that's combining with the carbon that's already in the cycle, and we are adding in other intermediates, and ATP has to be used here. This process of making sugars, we're binding things together, we're making bonds. We've got to have some form of making more sugars. And so the way we are doing this is by bringing those carbon dioxides in, breaking them apart and rearranging them, but using up ATP at the same time. So this ATP came back here from photosynthesis. The cell is using it to make this next step. Then we are also using the NADPH. We made that over here too. We're using NADPH, and we got that NAD+. This can go back into the first part, the first step. So we can use this again. Cycle goes around. Glucose is eventually going to be produced. Cycle keeps going. More ATP is used. In this case, it's resetting. We have to reset the cycle so it's ready for the next start. And that's what these ATP are doing. It's resetting the cycle, getting it ready to start again. So to summarize with this, we have ATP coming in, NADPH coming in, carbon dioxide coming in, and we have NADP being produced, NADP plus being produced, and sugars being produced. So 3, this is called a PGA, is reduced to a BPGA. Again, it's all kind of confusing, but for our purposes, just really try and focus on what goes in and what comes out, the reactants and the products. And we do need to reset the stage after all the materials have been used up. The NADPH and the ATP, these were all acquired from the light reactions of photosynthesis. This is kind of showing how it works, where you use that ATP to make ADP and use that NADPH to create NADP+. This will go back to photosynthesis, and this will also go back to photosynthesis, or this can go to cellular respiration. Ruby-P is used in CO2 fixation and must be replaced every 3 turns of the Calvin cycle. So we're having to replace things periodically and reset things. So 5 of these is going to make 3 of these, and Phosphates, again, are being formed because we're having to break up those ATP, and this is the resetting phase. Toward the end of this cycle, you have to reset it and get it ready for the next step. Glyceraldehyde 3-phosphate can be converted to many other molecules. It's abbreviated G3P. We see this in fatty acids and glycerols, glucose phosphates, fructose, starches, and cellulose. So these things can be used for other purposes. So here is just kind of showing that as a tree or a plant in general uses this G3P, it can be turned into these different things that also have multiple purposes and can be used in this case to the leaves or the roots or the cellulose. So there are other types of photosynthesis. In hot, dry climates, the stoma must be closed to avoid wilting. We mentioned that the stoma, when it's open, it's great to get carbon dioxide, but sometimes that's not helpful because the water is evaporating quickly and the plant wants to save water. When this happens, it decreases CO2 and increases the amount of oxygen that's being produced because you are still using what water you have, and that's where the oxygen comes from. So oxygen starts to combine with RuBP, leading to the production of carbon dioxide eventually, and this is called photorespiration. So this is something that plants will typically do in places that are very, very hot, like desert climates, and these are called C4 plants. So they solve the problem of photorespiration by fixing that carbon to PEP, which is a C3 molecule. So basically, C4 plants are better at operating with their stomas closed, or stomatas closed, and C3 plants are your typical plants that you like, grass and trees that we're familiar with. But C4 plants are better at using this process and keeping their stomatas closed. But if you put them in a cool, moist environment, C4 plants aren't going to do as well as C3 plants. They're just not adapted for that. So here's a C3 plant and a C4 plant, and you can see there's lots of different arrangements in their cellular structure. So we've got bundle sheath cells and the stomatas and a vein, which allows for the movement of materials, and it's arranged very differently here. And this is going to protect the very vital things that are going on the inside and making it so that they are capable of obtaining the nutrients that they need even under very dry conditions. Carbon fixation in C3 and C4 plants. So here is a C3 plant, and you can see just kind of how the basics work. Here is a C4 plant that has a little extra step here with the mesophyll that is aiding this process and making them a little more efficient. Now there's also something called CAM photosynthesis, cross-Lucian acid metabolism. CAM plants partition carbon fixation by time. This is if it is just way too hot during the day, but it's better at night. So let's do some processes at night and some processes during the day. So CAM plants fix that carbon dioxide at night because it's cooler and they can open their stomatas at night. So when their stomatas are open, they're not losing as much water and they're able to fix that carbon dioxide better. And during the daytime is when they're going to they have that NADPH and ATP available. The stomatas are closed during the daytime, but they're able to still complete some of that processes because they've relegated some of the process to nighttime instead. So here's an example of a pineapple, which is a CAM plant. You have the carbon dioxide coming in at nighttime when the stomatas are open, C4 fixation and carbon dioxide, and then during the daytime you can do the rest of this process because you don't have to have your stomata open because you did that when it was cooler. So photosynthesis has plants that have adapted to their particular environments, and this just shows how some plants can adapt better and are better at living in certain conditions. C4 plants are mostly adapted to high light intensities, high temperatures, and very limited rainfall. So again, think like a desert. C3 plants do better at colder temperatures, so those are the ones mostly around most of Missouri and most of the United States. Cam plants are better adapted to extreme aridity. There are flowering plants that are found in these conditions, and there are some that are found in non-flowering plants as well. All right, so hopefully that helped with photosynthesis. It may be to your advantage to watch, especially the photosynthesis and the light-dependent reaction. Watch that a couple times, or like I said before, going into your YouTube and watching different videos on YouTube that help explain the process, because in those you can actually like see the hydrogens moving and see the electrons bouncing because they're animated. So if you have any questions or any concerns, please let me know. Bye guys.